Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speakers, Danny Spurgeon and Cassidy Marquis from Repligen.
Thank you, Leah. This is Danny. I'm a product manager here at Repligen. I want to thank everyone who's attending. Very encouraged by the numbers of people. Thank you for joining us. I hope you find that it's useful. We noticed a lot of you are filtration, but we also have some dialysis friends and family, so I'll do my best to show both sides of that coin. So today I'm very excited to talk to you about dynamic dialysis, a novel method for process scale membrane separation. So in this short webinar, we're going to go over a few things. First, we're going to talk about and refresh you on the basic principles of dialysis, talk about what you may be used to as the traditional static approach, introduce the concept of dynamic dialysis, go over briefly Repligen's portfolio of dynamic dialysis systems, do a little bit of PD, proof of concept experiments. At that point, I'm going to pass it over to my colleague Cassidy, who's going to go over some practical applications, wrap up, and we'll have hopefully plenty of time for Q&A. So let's start at the beginning. Let's contrast filtration and dialysis. So filtration separates under pressure. Think of a French press when you're making coffee. Filtration is quick, but it will not work in all purification. Dialysis is a gentle, no-pressure separation driven by concentration gradient. I have that underlined because that's the driving force that I go back to throughout this presentation. Think of tea diffusing into water. It will purify most compounds. However, the trade-off is it is not as fast. So traditionally, dialysis is used at the research analysis scale, and at scale-up, many companies will switch to a pressure-driven technology like tangential flow or normal flow filtration. The crux of this presentation is I want you guys to consider dialysis as a large volume separation tool for downstream purification. It's specifically good in niche applications that may fail under pressure-driven filtration. There's many different places, but the top three would be viscous samples, things that are simply too thick to pump through a filtration, a labile protein if you have a protein that's subject to shear, or a hydrophilic gel matrix which would form a gel across your hollow fiber filtration membrane. So let me walk through this little cartoon of traditional static dialysis. So this drawing, you have a beaker. Blue would be your buffer. This is your dialysis membrane. The orange is your retentate, what you want to retain in your sample. And the green is the contaminant, what you're trying to eliminate. At the initial point of this dialysis, you have a very good concentration gradient because there's no contaminant yet in the buffer. So that difference is going to be what is going to drive the contaminant from inside the membrane, passing through the membrane into the buffer. Because there's a drive towards equilibrium, this reaction is going to slow and eventually stop when there is an equilibrium between the concentration of contaminant in the buffer and within the membrane. In order to reset that concentration gradient, you need to manually put fresh buffer in, and your reaction can continue. While this works for many people at the benchtop scale, it gets increasingly and exponentially challenging when you're dealing at large-scale processes. First of all, it's an open system. Requires manual buffer changes, and when you do those manual buffer changes, you have to physically handle your membrane. So if you have a valuable final product in there, it's subject to potentially ripping, which is something you want to avoid. It's also limited in scale. Generally speaking, you use 100 times the amount of buffer compared to the size of your sample. So when you're dealing with multi-liter samples, it gets very onerous to try to change that much buffer. Because of this drive to equilibrium, static dialysis has a low separation efficiency. It requires labor time. We have one customer who has to pay a research associate to come in at 2 a.m. to do a buffer change. Very inconvenient. So this is dynamic dialysis. We feel it's just a better way to dialyze. You have a reservoir of buffer here on the left, peristaltic pump, a dynamic dialysis system, and a waste container. The peristaltic pump is at a very low flow rate, constantly flowing buffer over the sample. This does two things. One, it helps remove the contaminant from the sample into the buffer, and it immediately expels it to waste. Constantly keep upholding the maximal possible concentration gradient. There's many benefits for this, especially at the large GMP scale. First of all, it's a closed system. Secondly, you have that continuous buffer change. There's no need to manually change the buffer. There's also no in-process membrane handling. You're not putting your sample at risk. You put your sample in at the beginning of the reaction, and you pick it up at the end of dialysis. It's very easily scalable. All you need is more buffer and more membrane. Because the contaminants are immediately expelled to waste, you have maximal membrane efficiency throughout the entire reaction. And it's a walk-away convenience. You never have to come back to babysit. So these are a few of the product offerings that Repligen has in dynamic dialysis systems. This one here on the left is our 10-liter tank, which is used to process up to 2-liter samples in dialysis membrane. The minimum working volume for this is 250 mils. The reservoirs here, the tank, the tubing, and the waste tank are all made of GMP-grade materials. And we also offer a 5-liter tank and a 7-liter tank for smaller samples. On the right side, fall of 2018, we'll be launching a much higher scale version of this, in which we have six 10-liter tanks operating in series off a single reservoir and a single peristaltic pump. Using these 6 10-liter tanks, you can process 12 liters of sample in a given batch. So now I'd like to take you through just a few proof of concept and PD experiments. First, we're going to take a look at comparing static dialysis to dynamic dialysis, looking specifically at the passage of vitamin B12 dialyzing against DI water. Now we use these scaled down devices. These are not commercially available to sort of amplify the effects and the differences between the two. We compared static dialysis to dynamic dialysis at a flow rate of 6 mils per minute. It was a 1-hour experiment. We were just looking at the initial passage of vitamin B12. It should be noted that in static dialysis, you usually don't do your first buffer change until 4 hours. So you can see after our experiment, we have here on the left, static dialysis after 2 hours, and here on the right, dynamic dialysis after 2 hours. The buffer chamber here, if you can see, there's sort of a pink hue to the buffer, which is indicative of the vitamin B12 buildup in the chamber. This is a low concentration gradient. However, on the dynamic side, we successfully expelled that contaminant to waste, and you're upholding a high concentration gradient. Our hope and expectation is that this concentration gradient would lead to a faster rate of dialysis, faster rate of passage. And that seems to be the case. Here, this graph, the blue represents dynamic dialysis, whereas the orange represents static dialysis. And dynamic dialysis has a faster rate of passage, initial rate of passage, and you can see that static is slowing towards equilibrium earlier. So if we determine that flow has benefit on the concentration gradient against static, what if we took a look at different flow rates? Keeping all other things equal, will we see an increased concentration gradient or passage of vitamin B12? So in this experiment, we used 3 scaled down devices, repeating 6 mils per minute, doubling to 12 mils per minute, and going 10x at 60 mils a minute. Again, we were looking at just the passage of vitamin B12, and we measured this over 90 minutes looking at that initial rate of passage. And for this case, we found that a faster flow rate is indicative of a faster passage of vitamin B12. You can see here in orange, this is our 60 mil per minute, and it led to the fastest flow rate. In our case here, we're using DI water, so using 10x isn't too much of a big deal. Further PD would be needed to find the optimal flow rate. Optimal isn't always the fastest, especially if you're dealing with a buffer that is onerous to make or something that's just costly. So you may want to find your optimal low flow rate to try to conserve buffer. So I hope that explained the concept of dynamic dialysis, the systems we have to offer, and sort of the PD that is needed to get a process underway. Now I'm going to turn it over to Cassidy, who's going to go over some practical applications.
Thanks, Danny. Hello, everybody. My name is Cassidy Marquis, and I'm the director of the dialysis division and have quite a bit of experience with the dynamic dialysis process application and development. So I look at a model of a dynamic dialysis application to help you understand how this translates to you in your practical application. As Danny had mentioned, every application is going to be different, and every application will need process development. So it really is a good place to start with a model and then go from there to fine-tune it to your particular application. So in this case, we're starting with a GMP batch production of a delicate protein that cannot be filtered because it would just simply be damaged through filtration stresses and pressures. So what we're doing is we're starting with an 80 kD labile protein, and we want to purify it to eliminate urea and low molecular weight contaminants that are less than 1,000 Dalton. The sample volume that we're starting with is going to be 6 liters, and we're going to take this sample and we're going to process it through our dynamic dialysis system. Starting with a dialysis membrane, in this case we're using our standard regenerated cellulose with a 12 to 14 kD molecular weight cutoff and a 75 millimeter flat width. This particular membrane we can customize specifically for the application to a pre-cut length. We can pre-irradiate it, and it will be packaged specifically for GMP manufacturing for that batch quantity. The system in this case is going to consist of 3 10-liter tanks connected and operated in series. So we're going to have a single buffer source that's going to flow through a pump into an initial tank that will hold 2 liters. This will continue a second tank that holds a second 2 liters, and then into a third tank that holds another 2 liters. So for a cumulative 6 liters of sample that can be purified. Then the waste is simply sent to drain. So this is the system, and then the operation of it, we're going to flow at 100 mils per minute. So you don't need to have that flow rate too fast and unnecessarily spend buffer. You want to optimize the use of your buffer and conserve as much as you can. We're going to operate for approximately 16 hours, and after 16 hours we will consume approximately 96 liters of that buffer. After 16 hours, when you harvest your protein, you'll find that you've removed about 98% or greater of the impurities. You're going to have a near total recovery of the protein. And because it's a much more gentle process than the other technologies, you'll see that there's a 95% of protein activity retaining. So once you've established a model of your process, then the next step would be to optimize that. And that is to go back and you want to fine-tune various parameters of the process to get the end product you want, increased efficiencies, and fine-tune that duration specifically for your process. And this is an example of an efficient technology scaled up for large volume manufacturing. So after looking at that model, then what you need to do is consider what applications would this be most suitable for. Well, it's really where filtration fails. And those are specifically going to be in shear sensitive products where they cannot handle the pressures and the stresses of pumping or filtering over a membrane. Some of the examples would be labile proteins, as in the case of the model I just explained, where you want to remove salts or low molecular weight impurities. This would be done for drug manufacturing. Also virus and virus-like particles also are fragile, and you might want to remove the glycerol, or you might want to do a simple buffer change for viral vaccine or various therapies. Also in drug delivery systems, when you have an encapsulated drug in a liposome, you may want to remove the free drug, maybe urea or even detergent. Another area that's specifically useful for dynamic dialysis is when you have a high viscosity sample. Either it's a high viscosity fluid or more even like a gel. So is cesium fractionated protein, where you're trying to separate protein in a cesium gradient, you may need to remove that cesium. And this is often done for immunotherapy proteins. You also may be wanting to purify a biopolymer gel that's extremely viscous and remove some of the undesirable copolymers, or you may want to remove some of the reagents that were used in that polymer synthesis. And then in the third area that it's particularly useful for dynamic dialysis is when you need to do low molecular weight polymer purifications, especially in synthesis, when you're trying to produce a polysaccharide, an oligonucleotide, or a polypeptide. And the things you might want to remove would be a monomer, or amino acids, nucleotides, or you may want to remove organic solvents or reaction solvents, or put it in a finished filled product. This would be done specifically for oligonucleotide synthesis, protein expression, or even in tissue engineering. So in these applications, dynamic dialysis is the preferred method over filtration because the filtration simply cannot operate for viscous fluids, or it is too damaging on fragile product. So in summary, dynamic dialysis is really a very good alternative to filtration in bioprocessing when you're dealing with some of those more specific areas of bioprocessing. Dialysis is stepping out of the laboratory arena and into the bioprocessing arena in the form of dynamic dialysis for GMP manufacturing. Repligen is the only company right now providing turnkey solutions and dynamic dialysis systems that are being installed and used globally for GMP manufacturing of biologicals. Culminating, the large volume dynamic dialysis is advancing the bioprocess industry. Repligen is available here to support process development and optimization efforts for your dynamic dialysis. So here's our contact information. If you need to reach out to us, we'd be happy to help you for these efforts and with your dynamic dialysis. And at this point, I'd like to thank you all for attending, and I would like to entertain questions that you may have.
Great. Thanks, Cassidy. So you offer closed systems and reduced sample handling. How are membranes prepared for use?
So the standard membrane products are not always best suited for GMP manufacturing because they come on bulk rolls that are either 15-meter or 30-meter length. And if you have to cut off a piece to use for a first batch, then you have a remaining amount that needs to be used in subsequent batches. That's not ideal in GMP. So the idea of customized membrane specifically for batch operations, it can be pre-cut to your specific sample volumes. For example, a liter volume sample, you would want to put that in a pre-cut 1 meter length membrane. We can pre-cut that for you. We can pre-irradiate that for you. And we can seal that in specific pouches that you would open at the time of GMP manufacturing to maintain that the package barrier up until the point of use.
How do you estimate the impurity removal based on the sample volume and the amount of liters passed through the system?
It's quite difficult to estimate the amount of materials that you need to remove, and how long it will take specifically. So the best approach to that is to start with a longer period of time. Let's say maybe 24 hours or 15 hours, and then work backwards. And the reason is every application is different and unique, and many different parameters factor into that. So things like, what was your sample volume starting? What is the concentration? What is the total mass of impurities that you need to remove? Also the molecular weight cutoff, the buffers that you're using. So all these things play into how long it'll take and how much you can anticipate removing over a specific time. So it's always best to start with a longer period of time and then optimize working backwards. And we're here to help you do that.
Great. And for viscous polymer solutions to remove small contaminant, can you discuss the pros and cons of this technology along with TFF?
Yes. So for viscous fluids, especially for gels, they really are not, filtration is not suited well for that type of a purification. You simply cannot pump viscous fluids or gels often across a membrane and get the separation that you need. For one, it would require significant pressures to do so because of the viscosity and can really impose significant pressures and stresses on the product, which can damage it and result in low recovery of your product or activity. Also, even extremely viscous fluids like gels, HA as an example, cannot be pumped. Instead, it's better to put that, encase that into a membrane that remains static, if you will, and then you actually pump the buffer on the outside of this encased sample to remove the solutes as they pass across the membrane. So the sample remains protected while the buffer is actually flowing and removing the impurities. This simply can't be done by filtration, or if it can, then it's not very efficient and can result in low recovery.
Okay. And what kind of solvent can be used in the dialysis reservoir?
The dialysis reservoir is primarily composed of a polysulfone plastic. There are some adapters that are polypropylene. So it really has a very good chemical compatibility for most solvents. And those plastics specifically, we have information on our website that you can look up for specific chemicals. But all in all, most acids, bases, and many of the organic solvents can be used with these tanks. Really, what it boils down to, though, is the membrane is really what would limit the chemical exposure. The standard regenerated cellulose, it does have a good chemical compatibility. However, it may not be compatible with some very strong solvents like DMF. So it can be used for concentrated weak acids and bases. It can be used with dilute strong acids and bases, and it actually can be used for quite a few different organic solvents, but those are the things you need to verify with, either with us or it's available on our website. So the regenerated cellulose membrane really is the benchmark for where you need to establish your compatibility for your process.
Okay. Well, I think that's all the time we have for questions right now. So thank you, Danny and Cassidy.
Thank you very much.
The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Thanks. Goodbye.